Hello everyone. Thank you for this opportunity. Today I am going to talk about for the generation of hypothetical models of new proteins identified in the venom gland of transcriptomic of Bothrops asper and Bothrops jararaca. My name is Joseph Sping. I am master student at computational biology in Pontificia Universidad Católica del Ecuador. This research was conducted with Doris Vela. For the introduction, the snake venom is a complex mixture of proteins with potent toxic effects. Among the medical important snakes in Latin America are Bothrops asper, found in Mexico, Panama, Colombia, and Ecuador, and Bothrops jararaca, endemics of Brazil's Atlantic forest. While the many venom proteins are well studied, but the low abundance, but the low proteins are unexplored. It's important because it could reveal new aspects of the venom functions. The objective of this research is model new proteins identified in both of the species. Um, in this case, use a reference sequence for the different species like wasp, ants, and cobras, and focus in the proteins for allergen 5, bone withering factor, arisulfatase, botrocetin, dihydrotate dehydrogenase, and basparin. In the method, the venom gland transcriptome of Bothrops species is assembled with Trinity, and its assay is used to modulate the proteins and validating with Ramachandran plot and a rat. Finally, PFAM help us to identify the conserved sites and functional domains. In the results, we identify all 6 target proteins and divide in new isoforms and neoproteins. In new isoforms, in both of the species, like we identify allergen 5, in Bothrops asper and Bothrops jararaca have the same conserved site with the reference protein. The reference protein is for the Vespula germanica. It is a wasp. Both models have the same conserved site and a unique structure, alpha beta alpha. This structure is important to improve the analytic effects. Another protein, we identify botrocetin. This protein have a one only record in UniProt database, and the structure is conformed with 2 chains. We focused in chain B because it can bind with the bone withering factor. Bone withering factor is a protein in mammals, and it's important to regulate the coagulations. Um, in Bothrops jararaca have the same Z-type lectin domain, but a different conformal structure. But a different structure. But on the other hand, Bothrops asper is have the Z-type lectin family, but it is most closely related to asparcetin B chain. Asparcetin is another protein hemorrhage, and have functions to bleeding the victims. Both models, Bothrops asper and Bothrops jararaca, from to the same family protein. This is zeta-lectin. This family is important to remove the bleedings and necrosis. In new proteins, we identify bone withering factor type D. The primary structure for these proteins conformed with beta sheets, and in Bothrops jararaca is the model full recovery to the reference protein. Is Odomatus monticola is ant, and have the domain bone withering factor type D. In mammals, bone withering factor regulates the correct coagulation, and it's probably that the snakes have a homologous protein with the same role. But however, it's also possible that the bone factor in Bothrops jararaca in inability disrupt the correct functions of the bone wear factor in mammals and promote bleedings and other effects. Basparin is a protein identified in Bothrops asper. It have a one record in UniProt database and NCBI. This we recorded the conserved zinc binding. It's important to establish the structure for this protein and connect with another proteins and promote necrosis. The Bothrops asper model have the same disintegrating domain, it's in green, but to the reference have another orientation. It's possible because this protein have this family disintegrin is conformed for the metalloproteinases and have a diversity proteins. It's very diversity. um, Arisulfatase, in this case, is the first result for this protein in Bothrops species. In snakes, arisulfatase is not studied, and one record in the lapid, but in viperinates not have records. Both models, Bothrops asper and Bothrops jararaca, have a stable structure and have a conserved site sulfatase. The sulfatase motif is important to degrade the glycosaminoglycans. Glycosaminoglycans is a structure rich in sulfatase and conform different like tissues, tissues like ligaments and vascular endothelium. This protein, arisulfatase in Bothrops asper and Bothrops jararaca, was suggesting that possible to degradation tissues during envenomation. Finally, dehydrogenase is a mitochondrial protein. This stable structure is conformed with 12 alpha helices and 8 beta sheets. In models we recorded have a stability structure with a dehydrogenase domain. This mitochondrial protein is the first result for this class of protein because it's possible that supporting the metabolic demands for the venom productions. This area is not studied, and this is the first class of this protein in these effects. In conclusions, the hypothetical models generated for these proteins demonstrate stable structure comparable with the reference proteins. This stability structure highlight and your possible functions, highlighting the importance of the low abundance proteins in the venom, and it's important to study this area. In another hand, these methods for the using computational methods, for example, it's asper, is a good alternative for this low protein because identify for the traditional methods in method labs, it's possible to might miss another proteins. Arisulfatase, arylagin 5 is not possible to identify with with traditional lab methods, but is possible to study your functions and your stability, your structure in computational methods, for example, it's asper or AlphaFold. Thank you very much. I am.
Thank you very much. Do we have any questions? Guys that are leaving, please, we're going to take a picture. So if you can wait just a couple of minutes, I will appreciate it. So, what? Oh yeah, do you have a question? Okay. You can go. You can come here and turn on the microphone, please, lady. This one.
Okay. Thank you very much. Your presentation is amazing. Nice talk. Just the existence of Bothrops in Peru. Are you maybe did you want to compare your results with any results about Bothrops in Peru? It's just a question to know more about your studies comparing with studies in Peru. Thank you.
Okay. Bothrops species in Peru? Yes, because we focus on Bothrops asper. This snake is interesting because have two distributions for the Occident and the Orient. We focus in the Occident zone, but in Peru the zone Occident is very desert and it's not possible to find these Bothrops. But in the Orient, way yes, have a different species of Bothrops and I read an important research for the Bothrops in Peru. The most important species in Peru is Bothrops. Bothrops atrox is another species. It's another species, but it's different for the Bothrops asper. Phylogenetic in phylogenetic is separated with into groups, and
Congratulations.
Thank you. So I want to. Oh, another question there. Okay.
yeah, for the first time I use AlphaFold, but AlphaFold is very quickly, but in the models not have different comparations, but each Acer have models is during 3 or 4 days, but this method is very lowly, but have different models to compare with the generate model. Um, I use AlphaFold to compare the models for generate with each Acer, and it's the same models, but the difference is in the validating. Each Acer validating the models with different techniques. One other is factor B, and it's important because validating the models is important to view the stability structure, but AlphaFold no have many validating. Yeah. Que le vi que estaba media sorprendida.